they are actually compounds that have a very specific structure associated with their ability to harvest light energy. And many times they also have structures that enable them to associate tightly with membranes. So they have a hydrophobic component that's able to localize within membranes. So these are, and their job is to, there's a whole set of these, and different pigments are able to absorb different wavelengths of light. And the wavelengths that are not absorbed are reflected or transmitted. So the reason that we observe plants and leaves to be green is that this is the light, these are the visible wavelengths of light that are actually not being absorbed by chlorophyll and related pigments in plants, right? So they're actually not utilizing those green wavelengths. They're using light that's on either side of that. And this is just, again, showing what happens. So we have, here's again, this cartoon of the chloroplast where light is coming in. And of course, there's a whole spectrum of light. And it's only the wavelengths that are on the relatively the far ends of the visible spectrum that are actually being utilized. It's the wavelengths in the middle, namely the green part of the spectrum here, that is actually transmitted or reflected, not absorbed. And so that's why we observe plants to be green. So let's take a little break and do one question. So pull out your clickers. Okay, so take about a minute to do this one, and I'm going to start the timer right now. Okay, so about 15 seconds. Okay, 5 seconds. And stop. Okay, that was kind of an easy one, right? I set you up for that. So 92% of you said that the answer is D. Okay, the answer is D. Uses all of the visible spectrum for photosynthesis. And that's right. If you see a plant that's black, you know that it's basically absorbing all of the light in the visible spectrum, and it's not transmitting any of it. So the plant was black. What if the plant was white? The opposite, right? The opposite. Everything's being reflected or transmitted. Nothing's being absorbed. And you could imagine other colors, different colors, potentially. Okay, so what we can do in the laboratory is we can try to figure out how plants are doing this using various kinds of devices. And I know that those of you who are taking the lab class will know about this, that we can use a spectrophotometer that measures the ability of a pigment to absorb different wavelengths of radiation. And this is a machine that, and we're talking about visible light here. So we're talking about a machine that is able to send light through various pigments that we're testing and measure the fraction of light that is transmitted at each particular wavelength. So it's a convenient way to try to figure out for a particular pigment or solution, or any kind of sample really, what radiation is getting absorbed and what radiation is getting transmitted or reflected in the process. This is a cartoon that shows, that's taken from the book, that kind of shows how this works. And so the idea is that you have a source of white light here, so we have all of the visible wavelengths represented, that come in and they interact with a refracting prism that breaks these wavelengths down into their individual wavelengths of the visible spectrum. And they pass through a slit so that we can actually adjust the slit to just focus on one part of the visible spectrum. And then this light that gets through the slit of a particular wavelength then interacts with a sample that's over here. And in this example, they're showing you a sample of chlorophyll. And by the way, so thinking back to when we talked about cell fractionation or tissue fractionation, how did we get that sample of chlorophyll in the first place? Anybody know? Like grind up leaves or plant tissue, right, and do a fractionation experiment where we're fractionating different molecules that are present in there. When we look at the structure of chlorophyll, you can think about that question again in terms of how would you actually extract it, right? So would you use something that was hydrophobic, hydrophilic? We'll come back to that. Okay, so we have a purified sample of chlorophyll over here, and we're passing light through it. And if we pass visible light that's green in this wavelength here, pass that through the sample, and then we have a photoelectric tube that is able to generate a signal in response to light that's actually getting through the sample, if any is getting through. And so in this case, because green light is not absorbed by chlorophyll, it's actually transmitted. This light gets through, it actually strikes this photoelectric tube, we generate a current, and that can be detected over here, right? And so we get a reading on this little meter that tells us that indicates that chlorophyll is actually absorbing very little green light. And then, sorry? And then if we do the same experiment again, and we adjust the slit, so now we're looking at blue light, and that hits the sample, now most of this light is actually being absorbed by chlorophyll. So we don't get very much coming through, we don't get much of a current generated, and we detect that on this meter over here. So this tells us that chlorophyll is actually absorbing most of the blue light that gets through. So this is a very convenient way to do that kind of measurement to figure out what's getting absorbed and what's getting transmitted. Yeah. Why doesn't it just absorb everything? Well, it has to do with the chemical structure of chlorophyll, actually. Okay. So if you're asking a philosophical question, like why did nature do this? Why not use all the light, right? That's harder to answer. I don't know if I have an easy answer for that. But basically, if we look at the structure of chlorophyll, there's been lots and lots of experiments done to try to understand the chemistry of this, right? And what happens when light actually hits chlorophyll or related kinds of molecules. And so we understand that based on the chemical behavior and the way that those compounds interact with visible spectrum, they're actually not, electrons are basically not excited by green light, whereas they are excited by wavelengths on either end of that. Okay, so we can then plot things out. So we can plot out an absorption spectrum. So this is basically a graph that plots a pigment's light absorption versus the wavelength. And if we have an absorption spectrum of the major pigment in plants called chlorophyll A, what this shows is that wavelengths on basically on either end of the visible spectrum, namely blue-violet light as well as red light, tend to work best for photosynthesis, whereas chlorophyll A really doesn't absorb light in the middle of the spectrum, namely the green wavelengths. And then we can also plot what's called an action spectrum. And what this tells us is it tells us the relative effectiveness of different wavelengths of radiation in driving a particular process. And we'll look at an example of this in just a moment. So there mainly are three different types of pigments that we find in chloroplasts. Chlorophyll A is the one that participates most directly in the light reactions, whereas chlorophyll B is what's called an accessory pigment. So this is a pigment that has a structure related to chlorophyll A, but it's able to interact with wavelengths of light that are a little bit different. So it actually extends the amount of light, or the wavelengths of light that can actually be harvested by these cells and used. And then we have pigments that are called carotenoids that are called accessory pigments. And their primary role seems to be not so much harvesting light energy and using it, but rather absorbing energy that would otherwise be damaging to the cell. And so if you go to the health food store, you can buy carotenoid compounds to take, and you're told that these are good, somehow protective of your cells. I don't know if they are when you consume them that way, but that's the claim. And these are actually found in these are colored pigments. So if you have vegetables that are orange or red in color, beets or yams and things like that, these tend to be very rich in these carotenoid pigments, whose job is to absorb light that would otherwise be damaging to the cell. So let's look at what these types of spectra actually look like, because I think it's a little bit more informative to actually look at some data. So here's on the top is an absorption spectrum that, and in this case, these are separate spectra that are plotted for chlorophyll A, which is shown in the blue line right here. This is chlorophyll A. Chlorophyll B, which is in the green. That's this curve right here. And then for carotenoids, this line that's shown in orange. And we're plotting the absorption of light by these different pigments as a function of wavelength. And so what you can see when we compare these different spectra is that chlorophyll A is primarily absorbing light on the pretty far ends of the spectrum, right? So down here in the blue and violet wavelengths, as well as over here in the red, or sort of orange and red wavelengths, and really very little absorption going on in the middle of the spectrum. And then if we look at the spectrum for chlorophyll B, you can see that here this pigment is a bit different, right? It's actually not absorbing as much in these blue and violet wavelengths. It's actually absorbing more here, sort of at a slightly higher wavelength than what we see for chlorophyll A. So basically, when we have those two pigments together, they're sort of complementary, right? They actually extend the amount of visible light that can actually be usefully harvested by these cells. And then down here on the other end of the spectrum, the same is true. Basically, the chlorophyll B helps to extend the amount of light that can actually be absorbed in these cells. And then the carotenoids shown right here help to absorb some of the radiation that would otherwise potentially be damaging to the cells. What color is chlorophyll A? It's mostly green, right? Because that's the light that's actually getting through right here. Okay, action spectrum is shown here. So here we're plotting the rate of photosynthesis, and this is actually measured by the release of oxygen. So again, remember that that oxygen is the output from these light reactions, right? So we can actually measure the flux of oxygen output and figure out what is the rate of those reactions as a function of wavelength here. And that's the plot that's shown right here. So sort of reflective of what you see on the top with the absorption behavior of these different pigments. If you saw that, you sort of might be able to sort of predict this plot, which is that we see much more oxygen being produced at the far ends of the spectrum, either at the lower wavelengths here or at the higher wavelengths here, than we see in the middle, where most of the light is actually just being transmitted. It's not, or reflected. It's not actually being absorbed and used to drive those reactions. So we're not getting very much oxygen production here in the middle. So there's a really cool experiment that was done to actually test this directly. And this was an experiment that was done back in the late 1800s by Engelmann. And what he did was this was the first time that an action spectrum of photosynthesis was actually plotted. And he did this in a really cool way, a really kind of a low-tech but clever way that took advantage of what he knew about the behavior of different kinds of organisms. And so what he did was to expose different parts of a filamentous alga to different wavelengths of light. And so the nice thing about using this as a model system is that these alga grow as these long filamentous structures, so they're just physically elongated. You can stretch them out, and if you have a prism that's breaking visible light into its component wavelengths, you can then just expose different parts of this filamentous alga to different wavelengths of light in such a way that they're pretty nicely segregated from each other. And so that's what he did. And so, of course, the reason to do this, what he was trying to figure out was really asking the question, which wavelengths of light are most beneficial in terms of driving these light reactions. And light reactions are producing oxygen, and so he needed a way to detect the amount of oxygen being produced from these algal cells that were exposed to these different wavelengths of light. And so what he did was to use the growth of aerobic bacteria. So these are bacteria that require oxygen for growth, so they grow better when they're in the presence of very high concentrations of oxygen. And so he had these bacteria, it has sort of this alga stretched out, being exposed to different wavelengths of light. He had these bacteria plated out alongside this alga. And then just look to see where did these bacteria grow best along this alga and use that as a measure of oxygen production. Pretty cool, right? Pretty clever. And so if you look back at this picture right here, this is just showing what this experiment actually would have looked like. So you had this filament of alga and being exposed to these different wavelengths of light that are being separated out. And then he has these bacteria that are plated out. And what he could see was that these bacteria grew really well down here at the lower end of the spectrum, these shorter wavelengths of light. And then they also grew pretty well up here at the higher end of the spectrum with these longer wavelengths of light, and not so well in the middle, right here, where, as we now know, most of this light is actually not being used to drive the light reactions. It's being transmitted. So this was just a really direct way to try to get sort of indirect way, I guess you could say, of detecting where oxygen was being produced, and therefore understanding where these light reactions were actually most efficient at what wavelengths. All right, so I just want, in the last few minutes, I want to talk a little bit more about these pigments. So as we said, so chlorophyll A is the main photosynthetic pigment. And then we have these what are called accessory pigments, and chlorophyll B is an example of this that help to broaden the spectrum that's used for photosynthesis. And then we also have other pigments like carotenoids that help to absorb light in wavelengths that would otherwise be damaging to chlorophyll. Yeah. Can they absorb ultraviolet light? I think some of them can. Yeah, it depends on the structure of these compounds. And there's many of these, right? Not just one type. So this is actually the chemical structure of chlorophyll, and here you get a sense of, from looking at the chemical structure of how these are actually designed beautifully they sort of have evolved to have components of their structure that are capable of different functions that are necessary in the light reaction. So very importantly, there's a porphyrin ring up here that has a magnesium in the center, and this is where the actual light harvesting is going on. Light hits this, we get, as we'll talk about more next time, we get excitation of an electron that is then, when it decays, is able to, that energy that's given off is able to be harvested, or at least some of it is. So this is the head of the chlorophyll molecule. very interestingly, so the difference between chlorophyll A and chlorophyll B is just this very, what looks like a very minor change, right? One substituent right here on the porphyrin ring, but that actually has a big effect in terms of the wavelengths of light that will actually lead to excitation of an electron in the system. And then I also want to just point out that this has a hydrocarbon tail, so this is very hydrophobic. And this is important, as we're going to see next time, because this pigment is actually sitting in the membrane, right? It has to be associated with these lipid bilayers, so it's very important that it has a lipid tail that allows it to, or at least a hydrophobic tail that allows it to do that. Okay, so that's another important component of these types of pigments, is that they have a hydrocarbon tail that allows them to interact efficiently with hydrophobic parts of both proteins and also the actual lipids in the membrane. So what happens when chlorophyll interacts with light? So it turns out that when a pigment absorbs light, it goes from a ground state to an excited state. And this excited state is energetically unstable. And when excited electrons then fall back down to the ground state, photons are released. And we can actually detect this if we have this system uncoupled from anything else. In other words, if we have a purified solution of one of these pigments like chlorophyll, we can actually detect that decay of these excited electrons by observing fluorescence. So if we have an illuminated solution of chlorophyll, we'll actually observe it giving off both light and heat as those electrons decay. And so this is a cartoon that sort of shows what this looks like, where we have chlorophyll molecules that are interacting with photons, exciting electrons, and then when these electrons decay back down to the ground state, they do so with the release of both heat and photons that we observe as fluorescence. And that's what it actually looks like for the solution of chlorophyll. Okay, so on Friday, we're going to talk about then how that energy is actually used to drive the reactions of the Calvin cycle.